Next, I'd invite Dr. Shawn Hervey-Jumper to the podium. He's a brain tumor neurosurgeon as well from the UCSF, but you will all be very happy to hear that he has a much thicker cortex than I do, so your talk will be exceptional, I'm sure. Shawn, go ahead.
I have to say, I am so honored to be here. I don't think this many nice things about neurosurgeons are said at neurosurgery meetings. So I'm really, really blown away. It's very nice. Listen, I'm a surgeon scientist. I really come from a cancer biology background. And I'm going to talk through some of the things that we've learned or tried to learn with respect to neuronal innovation of gliomas. And let's think about it this way. I'm not sure we've understood that really fully the clinical context of what we're talking about in some of these disease conditions. This is a person I think about often. There's a patient who came to me. We clearly see the tumor, right? Standard of care treatment ends up with a tumor resection, chemotherapy, radiation, 2 clinical trials, a BRAF inhibitor, he was on a MEK inhibitor clinical trial as well. Ultimately, he succumbed to the disease, and his last MRI looked like this. The reason I put this up here is to remind me, maybe more than anything, that not so much the disease that killed him. This patient ended his own life, and he ended his life using medical approaches because the neurologic impairments that he gained throughout the progression of the disease were so devastating to him. Can't move my arm, then I can't move my leg, then I can't speak. So his final act was, I don't want to do this anymore. And so as a neurosurgeon who treats these patients, I will say, like, there's an incredible body of literature regarding this, and this has been summarized in a number of different ways, but just to put it in a couple of sentences, I've wanted to understand whether just the act of having cancer alone kills neurons, or there's something more to it. And the general, overall view that I'll put forth in summarizing, 20 years of work, starting with Carol Sontheimer's work in 2021 and 2001 with ion channels in cancer, and then Make and Nadergaard really putting glutamate on cancer cells and watching them grow, has evolved into this amazing field in which we now know that there's glutamatergic and GABAergic units, both directly innovating through bona fide electrochemical synapses, as well as paracrine-mediated things coming from malignant cells that make these cancers grow. And by the time that these tumors grow, then they therefore secrete other things that make neurons hyperexcitable in a number of different mechanisms. This has a role in cancer initiation, as well as tumor progression, invasion, as well as treatment resistance. And again, as the neurosurgeons in the room, when you think about this, are probably saying some obvious things. These are a list of a couple of people that come to see me in clinic, 19-year-olds, in the military on the far left, to a new college student on the far right, and everyone in the middle. And we understand, with one hat, that we really need to take out the tumor, remove it. But rarely is all the tumor removed. Even the grossly evident tumor is not removed in many contexts because of where it is or its behavioral significance. And so this idea that there's residual tumor in many cases, and for some patients, present in their brains for years, including decades, is something I wanted to understand. And because of this, it's been about a decade now trying to build an annotated data set to help us understand and break apart some of these mechanisms. So this idea, this annotated data set is now at this point about 750 patients, about 12,000 electrodes that are behaviorally annotated with various contexts to try to help understand this synaptic idea for IDH wild type and IDH mutant gliomas and what that significance is on tumor growth. I was really inspired by some of the talks over the last couple of days, and so I added some slides here, including some unpublished data that I think might be helpful. Much of what we're going to see here is based on this clinical annotation, right? This is somebody who's coming to the operating room for an awake craniotomy, no anesthetic, and a behavioral paradigm that determines that they're at their baseline relative to a preoperative testing set. We call this a wakefulness task. Then we align on the cortical surface using low and high density electrodes, now high density electrodes, each site that's removed. With the high density electrodes, we try to remove sites, tissues that are around 2 millimeters in size. It's called a micropituitary because it allows us to align spatially per electrode. We get between 6 and 20 of those per patient, and there are other cases in which we take larger areas of cortex for other experiments that we'll talk about. In this context, first set of experiments, that really was this that got me interested. And I'll buzz through these next couple of slides because it was beautifully described by one of the other speakers. This slide surprised me. With the idea going into this that cancer kills things, if you look at only cancer-infiltrated electrodes, this is a cross-subject analysis, looking at high gamma power, right, some basic crude measure of local neuronal synchrony, now looking only at cancer-infiltrated electrodes that I expect to just be garbage. It's just supposed to be noise. But unfortunately, we see at time point 0, this is a person saying a word, 800,000 milliseconds prior to speech onset, you see this increase in high gamma power within your Broca speech areas, which then suppresses after speech onset, to the point where your task-evoked responses or behaviorally congruent task responses not just elicit a high gamma power relative to an age-matched control or within subject non-tumor electrode, but it does that in a hyperexcitable manner. One point about tumor and non-tumor in this, because we've had a couple of glioma talks. I'm going to use a very pragmatic and simple version of what's tumor and what's not. It's based on visual inspection and based on flare infiltration. MRI shows flare infiltration, binary, this tumor or not tumor. It's very crude, but everything in that region is cancer. Everything outside of that region is probably also cancer, but with a smaller degree of cancer infiltration. But this platform or this system really allowed us to then assign for each electrode or using magnetic encephalography, each voxel, areas of cancer or glioma, cortically and subcortically, that were hyperexcitable, that then we can take those cells and run fun experiments with it. I'll buzz through this. We've talked about this already. I was very interested in these synaptogenic tumor-derived cells, again, based on this idea that there are spikelet membrane depolarizing malignant cells. I wanted to know whether they might be within these regions. We call these HFC cells. We co-culture these with various types of neurons. These are iPSC-derived neurons. We use this early. They're not electrophysiologically active, but they secrete a bunch of trophic factors similar to mature neurons, but we got away from these after a while. But using some of our better and different models, we could see that if you just take these highly connected intratumoral regions and look at the proliferation, and in these active intratumoral regions, you see an increase in proliferation. And oh, by the way, if you just take the conditioned media of neurons and add those to glioma cells from those HFC regions that don't grow on their own until you add either neurons or neuronal conditioned media using a proliferation assay or EDU proliferation assay, it makes those cells grow. And so these tumor-derived regions that are electrically or hyperexcitable are molecularly distinct, we believe. They proliferate in response to neuronal signals. They invade differentially, they kill the mice, they form these synapse-type structures. And we tried to apply these in a couple of different contexts. Again, for patients, if you use this connectivity concept to a patient's brain, now it's been done with DNA methylation, using connectivity measures, using subdural electrode array work. These patients with a functionally integrated or hyperactive glioma, IDH wild-type glioblastoma, pass away faster. And so some of the targets derived from these approaches are now working through clinical trials in various applications. There's about 4 or 5 of them that we won't go through for the purposes of today. What I did want to go through, and again, I changed my slides last night because of some of the talks the last couple of days, is this. So if we can convince ourselves that synaptic activity or tumor innervating or tumor intrinsic neurons are prognostically there and may be relevant, well, what is the landscape? What are they? What are these? And I was also really inspired by Dr. Palmer's work when that publication came out and as it was going through review. And so we just said, at a postdoc in my lab that came and said, well, is cortical laminar structure even preserved in the setting of cancer infiltration? Couldn't find a really good answer for this, but we thought this was a really good platform for spatial resolved transcriptomics. This is the 10x Visium platform. And I have to say, as a surgeon, I love how much context and confidence you guys have in each of the surgeons that you work with. I was pretty concerned that the results we would see were related to the way that I sampled tumors and tissues. So I was able to reach out to Vidi Ravi at University of Freiburg and Felix at Heidelberg, and I actually didn't tell them the experiment. I said, are you getting any cortical samples? Get your cortical samples from your surgeons. Let's come together and put this together. And it's just really an annotated data set now of 25 patients. Not huge, but something. We separated out into glioblastoma, as well as the 2 forms of IDH mutant tumors, really, right? Those who don't know or think about this a lot, there's really 3 categories of tumors in the adult hemispheric glioma we're thinking about. Glioblastoma, it's wild type. IDH mutant, and those are two, oligos and astrocytomas. One, the oligodendroglioma grows quite slow, and it's annotated by a 1p19q codeletion. And so we separated out the data into these three categories, and you can see characteristic examples. And then we could say identify excitatory and inhibitory transcriptomic profiles. But I really initially just wanted to understand cortical laminar order and whether that's even preserved. And this is where our beautiful Allen Institute atlases came into play. And we assigned a neuronal subtype for each spot, resolved at single cell resolution for a lamina, 1 through 6. Which helped us, again, resolve that you can identify laminar-specific changes here, and we can then separate them out by lamina into excitatory and inhibitory. And this is, again, using those same, 5 microns away, human tissue, and then look at proliferation from the upper cortical lamina, upper cortical layers down to L1 through 3, and then 4 through 6. And then we can identify, for the first time, the presence of this laminar specific. I called it a top-down synaptic enrichment. The reviewers didn't like that. They thought that had some other applications, so we call it a laminar specific synaptic enrichment that has a proliferation significance to that. Same thing happens if we xenograft these animals into xenograft these glioblastoma cells into a mouse. And these laminar annotations allowed us to do this. And I put this in last night based on some of the atlas data that we looked at yesterday. Because, again, actually seeing this data in a different light as a result of this meeting. You notice, again, I wanted to understand which neuronal subtypes are preserved and which are most vulnerable. And there are some real similarities between this and some of the Alzheimer's data that was presented, which really, again, has me wondering quite a bit with respect to how much of this is normal aging or not, or whether this is disease specific or not, or if it represents anything related to malignant cells at all, or could be just what we find in the setting of human brain and disease. So you can see now the breakdown between glioblastoma, oligos, and astros in terms of neuronal subtypes. So I called Eddie yesterday to tell, Eddie Chang is my neighbor and now boss and chair, and collaborator, and he's worked, as many of you in the field know, on neuropixel probe recordings, and I haven't seen much of that here because I don't think that's really the context, but I did want to put in a couple of slides on some of the preliminary work. Now, my lab is not a technology development lab. I'm definitely not a tech dev guy at all, but what I do want to understand is how active these units might be to then determine whether they're prognostically important or relevant and behaviorally responsive. And so let me walk you through a couple of slides for the data that's under review. So you can see the spike rasters, and what they're talking about are electrodes that are only implanted in flare-positive, flare-significant regions of tumor. So everything, again, using our pipeline, that is cancer. On the right, you'll see relative to the most age-matched control we can see, which is absolutely not a control, but an age-matched epilepsy cohort as best as we can find. You can see the clustering plots here, which we see both fast spiking and pyramidal type units, but this should eyeball really relatively well, which when you look at the epilepsy cohort relative to the glioma cohort that includes both IDH mutant and IDH wild type at rest, you can see here a reduction of units. Good, that tracks well. The excitatory units, we can see a reduction as well, but the inhibitory units shift in distribution, but the inhibitory units are still there, but in the lower cortical lamina. So the results of this, I think, are really important because using the whole cell patch physiology experiments and Harold Sondheimer's work, et cetera, there's really a suggestion that these inhibitory units were lost. I don't know, this will require lots of additional validation, but I wanted to share that in this context and this model. And a couple other slides just so you guys can beat me up over this later. The idea that these patients have significant behavioral impairments is something, again, as a clinically active neurosurgeon, is, I think, really important. So I really want to understand how behaviorally responsive these units might be. And so we thought about how to just break this down into maybe the crudest terms possible. So much of the clinical context for awake craniotomy for these patients is speech production and speech reception. They hear sounds or they say words, audiovisual picture naming. And so do we have behaviorally congruent neurons, both for production and reception? And you can see here again, just using an illustrative example, 3 neurons for production, a similar for speech reception. And I should also say that the electrode implant here are settings in which the tissue is going to be removed. So under clinical context, this is not tumor that's going to stay, this is tumor that's going to remove. So these are, again, not required for speech production or speech reception, but they still seem to be behaviorally congruent in some extent. This is a breakdown of what you'll see relative to slow-growing IDH mutant relative to the fast-growing IDH wild type, and that kind of tracks, I guess, with respect to the units. And you can see this is for 24 insertions and roughly 1,000 neurons. But this will just show one more slide here. I think about a lot that single units or single neurons really are not the cognitive construct we think about for patients. It's really things that fire together, right? And so this is a preliminary experiment that's, to see if we can even generate assemblies using this pipeline. And you can see here again, we can identify and generate neuronal assemblies for speech production and speech reception. I'll just show the slide here that shows the production versus reception assemblies. Much work to be done, lots of validation required, but I want to share that in this context to say that these are behaviorally relevant regions of cortex in the cancer that's going to be removed that might have implications for patients. When I found out I was speaking next to Lucy, I really wanted to add one more piece of this. Because if we can identify or really trust and believe that these neurons are behaviorally relevant and important, that really matters for not just high-grade glioblastomas. I would argue that maybe some of the biggest or largest concern that we have in a cancer field, are really patients with IDH mutant tumors. They're smaller in number, roughly maybe 20,000 a year, but they live for 20-plus years with their disease. And there's a new trial, it's called the Indigo trial, that came out, offers for the very first time a molecular therapy for these patients, but they all recur, even with our best drugs and therapies. This is our best therapy prior to the Indigo trial, which is focusing on the role of PCV chemotherapy together with radiation. And you can see how chemotherapy, this PCV chemotherapy, lomustine cocktail, improves survival, but these patients continue to progress. And this is my summary of most of the literature with respect to patients progressing. So these mechanisms by which glioma, these IDH mutant or slow-growing tumors, patients with a significant burden, by which they progress is really unknown, even with our best therapy. And so this is a nice collaboration, the functional validation on my side, the genomic side on the next-door collaborator side, David Riley, and he had just put together some of our samples from surgery and others over about 25 years for patients that had initial diagnosis and patient-matched samples for recurrence. And you can see here we ran this for single cell, single nucleus, and ATAC-seq. And you can see, again, this is a spatial map for our newly diagnosed versus recurrent samples, and I won't bore you with that. But we broke these patients down into observation, whether they receive radiation and temozolomide or just temozolomide alone. And you can see a nice, clear separation. And what you can see if you look at the differential gene expression plots is that many of the differential gene expression targets are synaptic in nature. And if you look at the ontologies for this, again, we're seeing things like synaptic transmission, regulation of neuronal projections, axonal development, and these are both, this differential gene expression plot or gene ontology is really focused on copy number alteration defined malignant cells. So if we then take this synaptic profile and say, well, what can we make any sense out of this? We developed a transcriptomic connectivity profile within these malignant cells. And of the 28 differentially expressed genes, here you can see the connectivity plots with separation between newly diagnosed and recurrent. This obviously requires histologic validation, and the major drivers of this seem to be TSP1, the tumor-derived synaptogenic target, NETRIN-G1, NDRG2, and connexin 43. Well, these were fun and interesting because TSP1 and connexin 43 actually have drug targets. And so we had to develop a model for this to try to study this better in a more nimble system. And anyone who's tried to implant IDH mutant cells into a mouse will know that these mice die of old age before they actually develop tumors. Tumors, maybe after 200 days, won't really take. And so we adapted a platform. This is a patient-derived organoid. So take patient-derived tissue, aggregate that, combined with either a mouse or a human neuron aggregate using E18 or P1.5 mice, or my lab is next to Arnold Kreigstein's lab, so we do have access to human cortical tissue, human fetal cortical tissue. So both of these work. After 2 weeks of the neuron aggregate, they become electrophysiologically active, and then you fuse these together with a V-bottom tube with some differential culture conditions. You can see, again, whole cell patch physiology suggesting mature neurons. We even you call this the shish kebab approach, showing that these are electrophysiologically active even post-fusion. We did obviously the immuno-EM to identify whether there are truly synapse-type structures in this context, and we could find them. And then we implant these onto either a low or a standard microelectroarray or a high-density MEA system. And you can see relative to our neuron aggregates alone, co-culture with our patient-derived glioblastoma cells in hypoxic conditions. I should say, I'm sorry, these have to grow on 5% oxygen, become hyperexcitable and proliferative, and invade into the infusion point. And you can see pre- and post-synaptic puncta expression as well as glutamate receptor expressivity that I won't show here. I want to end on this because the reason this is relevant is because in this model system now, if you drug for, again, a crude blocker of thrombospondin or a crude blocker of connexin 43 called meclofenamate, we can reduce the proliferation on the right, we can produce the hyperexcitability and the synchrony index. These aren't perfect, but it does open up a potential trial option that now is under development together with our targeted molecular therapies called vorasidenib. So I'll stop there, and I'm going to say, again, my take-home point is this. I think glial malignancies in the land where we talk about cancer, and this is kind of obvious to you guys, we talk about cancer as a cancer cell intrinsic mutation-derived system. These malignancies are electrophysiologically active, but behaviorally relevant in people. Both glutamatergic and GABAergic neuronal populations are present with some specific vulnerabilities depending upon the tumor type. It's new to me or new to us that oligodendroglioma evolution from a low to a high-grade disease seems to require synaptic integration. And this has actually gone through 3 rounds of revisions, and hopefully that'll come out towards the end of the summer. And I think this IDH mutant synaptic enrichment predicts clinical outcomes and is important. This is an amazing group of people that I work with and funding sources. We have incredible labs, maybe more incredible collaborators and students and postdocs, maybe more than anything. Really, again, as a clinically active person, really try this reverse translation approach. So I really, as much as I can, have to thank patients and families because without them volunteering for this, we wouldn't be able to do what we do. I'll leave with this. Again, these are 3 platforms that are humanized, right? Again, whether it's a neuropixel probe recording, that's, again, we can debate how valid that is and what that truly represents, or a patient-derived organoid with a GCAMP, calcium imaging for identification, or subdural array data. I think this is telling us things about cancer that we need to understand, and if we can understand that better, I think we can offer new therapies for patients. So thanks for the opportunity to visit with you.
Thanks, Shawn. Yes, over there.
Great talk. I was wondering about the VIP cells being particularly vulnerable in this case. Has any electrophysiological work been done to characterize their impact on circuit activity in the sense that because they're a major disinhibitory class of cells that primarily target other inhibitory interneurons?
Yeah, we have Catherine Cadwell her lab is around the corner from mine, and she's patching these for that intent to try to, she has a new grant through the Sontag Foundation to get to that. My take on it is probably, at least it's evolved a little bit over the last couple of days, but these seem to be like metabolically hungry cells, right? And as you might imagine, right, a cancer condition, there's a whole lot of new mouths to feed, right? These are, we have some work in that other paper that oxidative stress and oxidative phosphorylation is just through the roof in these regions. So it's probably just, oxidative stress and as part of the problem, but we haven't done the validation and kind of causally related experiments to get to that. But Catherine is absolutely working on that as an adjacent collaborator.
Thank you.
Hi, I was very intrigued by your neuropixel recordings. I was just wondering if you stick a probe into a tumor-infiltrated tissue, does the gliosis or the change in the physical extracellular matrix, do you think it changes some of the electrotonic properties of these neurons? Could it explain that you, I don't know, have a lower detection yield because it's more
Yeah.
insulated?
Yeah, it's possible. So I blame Eddie for this. So I was not going to present any neuropixel work, and Eddie's like, present the neuropixel work. So it's possible. I will say it's also possible that cancer just kills things, right? Like if I take a big step back, I kind of wonder, like, is this obvious that there probably should be a lower yield as well, right? The fact that you could identify transcriptomic profiles of neurons with laminar specificity was a little surprising to me, because I would expect that to just all be dead. But it's not, right? So it's your hypothesis is possible, and more work to do in that space to understand it for sure.
Thanks.
Any other questions? Shawn, I might ask you a question. Is there a point at which the interactions between neuron and glioma cells are just self-sustaining and we've missed the druggable or therapeutic window?
Yeah. So that's a really great question, and something we debated for about a year and a half. In the NCI, there's kind of three main therapeutic pipelines. One is the spore mechanism, and two are the clinical trial alliances. One is NRG Oncology, one's Alliance, for anyone who doesn't think about these things. NRG wanted a clinical trial concept for recurrent disease. I've read a lot of Shala Eroglu's work and others on the synaptogenesis inhibitors in general. My take on that literature is that most of the drugs and therapeutic approaches, regardless of disease state, do not do a good job dissolving existing synapses, but do a better job of preventing new synapse formation. Again, I don't know, many of you in the room are probably more experts in that space. I just read that literature. To me, that implies that you actually want a therapy after resection when the burden is low and you probably have fewer glioma neuron and synapses driving proliferation, and therefore you'd want to initial diagnose through recurrence. Problem with that comes is most neuroactive drugs have some amount of fatigue, right? What's the primary symptom that a patient with cancer has? Fatigue. They're all tired. So even if you use like any of these, clinical studies, Adam Sonnabend has this really nice focus ultrasound study where he applies focus ultrasound for blood-brain barrier disruption, of which these people have like relatively dramatic neurologic changes. Of my patients that I enrolled in that study for an implanted focus ultrasound device called Thera, 2 of them withdrew because they didn't like the fatigue and they didn't like even transient 48-hour changes in their nervous system. It made them feel bad. You know what I mean? So I don't know. That's part of the reason that the first clinical trials, I think, are really not just drug feasibility and biomarker based, but like, will people stay on the drug? I'm not sure.
Great talk, Shawn. Amazing stuff. Since you showed the neuropixel data, I have a question with regards to stability of those recordings. And
Say that one more time.
Stability, and to what extent you see a lot of movement. You have to do a lot of correction to. Basically, I'm worried about those waveform features, and to what extent those are robust, or you have to do something to massage them to sort of stabilize them.
Yeah, so this is where, again, I will say, and acknowledge again, we've gotten to the point where 2 people, I have a postdoc and a grad student in my lab that are integrated with Eddie's lab. I'm not in the technology development space. So the platforms and processing on that end is done with them. And so I couldn't speak to that. I shouldn't speak to that. I have some thoughts, but I really shouldn't speak to that.
Thank you.
Well, thank you very much, Shawn, and thank you to everyone who spoke at the session today. I know that both Lucy and Shawn said they do not like lunch, so if you want to ask them questions, they will be outside and ask them questions.